The development of beta-lactamase inhibitors seems to be expanding rapidly and promise to be the best short-term strategy for the most recalcitrant gram-negative pathogens. In this podcast, we will discuss the current state of the art with experts in the field. Welcome to Editors in Conversation. The objective of this podcast is to discuss how the discovery of beta-lactam inhibitors has evolved, review the current state-of-the-art development of novel beta-lactamase inhibitors, analyze the current and future clinical application of these drugs against major antimicrobial-resistant pathogens. This episode is brought to you by Antimicrobial Agents and Chemotherapy Journal, available at aac.asm.org. I'm your host, Cesar Arias, Editor-in-Chief of AAC. This podcast is also supported by the American Society for Microbiology, which publishes AAC. Do not forget to check our latest issue, fantastic papers available in AAC with papers on resistant pharmacology, antimicrobial agents, epidemiology, and clinical therapeutics. To discuss these important topics, let me welcome two experts in the field and real stars in antimicrobial agents. Dr. Robert Bonomo, who is Professor and Associate Chief of Staff for Academic Affairs and Director of the CASE VA Center for Antimicrobial Resistant and Epidemiology CARES at CASE Research Western University, and he is past editor of Antimicrobial Agents and Chemotherapy. And then Dr. Pranita Tama, who is Associate Professor of Pediatrics and Director of Pediatric Antimicrobial Stewardship Program at Johns Hopkins University, and is proudly an editor of Antimicrobial Agents and Chemotherapy. Welcome to the program, and thank you for accepting our invitation. So we're going to start with you, Robert, to give us a perspective of the field in beta-lactamase inhibitors. We've gone a long way since amoxicillin clavulanate, and the state of the art suggests that this field is probably the most useful to deal with really nasty infections produced by multi-drug resistant gram-negative infections. So give us a perspective of how these drugs have evolved through time, since you've really lived all of this.
Well, actually, they have an incredibly rich history when we go back and look. And even many of these papers were actually published in AAC. The first paper on clavulanic acid's discovery was, I think, in 1977 in AAC by Redding and Cole. And it's interesting when you go back to their original paper, you know, they tried a variety of different partners with the beta-lactam inhibitor clavulanic acid that they had discovered as a natural product. And that reading is fascinating. And some of the same approaches that we use now to evaluate our current new new beta-lactamase inhibitors was used even then. It took a while, but within the next year or so, sulbactam was discovered or synthesized, and tazobactam eventually, and they all took their place on the stage. And before we went online, Cesar says, you can challenge me any way you want in a conversation. So I will say you use that they're our best short-term solution. In fact, they may be our best long-term solution, because if you look back at the history of amox clavulanic acid, we're still using it. It's still one of the most widely available orally prescribed antibiotics. Pip-tazo, very widely described. And these drugs have had longevity because, for whatever reason, and it's part science and part good luck on our part, is that if you have a good inhibitor and you marry it with a good beta-lactam, you have a partnership that it's like a good marriage. It lasts forever. Well, sometimes. But, as long as it's pleasant, it's good. These are smart drugs. And I think, it's almost a logical development in the field that we went from these penicillin or beta-lactam-like inhibitors to what we call now beta-lactamase inhibitor, but structurally they're very close.
Yeah, the reason of the short term is something that I would like to discuss later in terms of development of resistance on these compounds.
Sure.
Before we go there, let's hear Dr. Tama So, Pranita, what do you think these drugs are so good to develop from the clinical point of view, and also feasible to develop at this point in time?
So thank you, Cesar, for inviting me and for getting to speak with Robert. I agree with Robert that I think as a class, beta-lactam antibiotics are, of course, the most commonly prescribed antibiotics, definitely the most clinically dependable antibiotics. And the development of resistance to beta-lactams understandably creates a lot of anxiety for patients, for clinicians, the scientific community, the public health community. And I think just a step back to get a sense of why beta-lactam, beta-lactamase inhibitors are so important, it's important to remember how they actually work. So just as a very simplistic overview, the bacterial cell wall is, of course, made of peptidoglycans, and cross-linking between peptidoglycan strands creates this mesh-like structure which provides strength to the bacterial cell wall. And it requires enzymes called penicillin-binding proteins for this to happen. And the way beta-lactams work is they enter the transpeptidase active site of penicillin-binding proteins. They mimic the terminal D-alanine substrate that penicillin-binding proteins bind to. And then they eventually form this acyl enzyme complex. It cripples the functioning of the penicillin-binding protein and eventually prevents the final step of cell wall synthesis. So obviously, this is not helpful for bacteria when this happens. So in response, they've produced beta-lactamases, which are bacterial enzymes that, of course, hydrolyze beta-lactam antibiotics, so rendering them non-functional. And as Robert was alluding to, when we think of beta-lactamases broadly, there's 4 molecular classes that are based on mechanism, conserved residue, sequence homology. These include the class A, C, and D, which have conserved serine-based mechanisms to hydrolyze beta-lactams. And the class B, we call the metallo-beta-lactamases, use a zinc-based mechanism. And I think that one of the reasons scientific progress into beta-lactam, beta-lactamase inhibitors has been so successful is they represent this established approach to reduce beta-lactamase enzymes, the activity of beta-lactamase enzymes, and therefore preserve the activity of penicillins, cephalosporins, aztreonams, and carbapenems. And I guess just very briefly, maybe I'll just state that the 2 bigger groups, and I think we'll probably talk about this later as we get into some newer agents. When we think of beta-lactamase inhibitors, as Robert mentioned, with clavulanic acid, sulbactam, tazobactam, we broadly think of them as suicide inhibitors. So they form these stable acyl enzyme complexes. They can undergo these post-acylation steps and eventually very slowly deacylate. And this is in contrast with the newer beta-lactamase inhibitors we have, which are reversible inhibitors. These include the DBOs, which stands for diazobicyclooctanes, but Robert can correct me if I got that wrong. So common avibactam and relibactam are the 2, and the baronates, vaborbactam. And both of these, so all 3 of these are unique because they can deacylate from the beta-lactamase without being modified, which is very advantageous because then they can actually go on and inhibit other beta-lactamase molecules. And I guess the last quick thing I'll say based on your second part of your question is asking why so much resources, I guess, are invested in beta-lactamase inhibitors. We, of course, before and ongoing, there has been investment and scientific discoveries in other beta-lactam compounds, notably siderophore antibiotic conjugates. Some of the previous candidates included monobactams conjugated with siderophores, cephalosporins conjugated to siderophores. But really, except for cefiderocol, which is currently FDA approved, none of these other previous candidates have gone beyond phase 1 studies. And this is thought to be mostly due to something called adaptive resistance, where microorganisms have this ability to rapidly adapt to the presence of an antibiotic, resulting in loss of effectiveness of the drug. And we think that the reason some of these other siderophore conjugates might have failed is because there might have been downregulation of iron transport receptors due to competition with the native siderophore population. And this might explain why some of these previous candidates looked really good in vitro, but unfortunately in animal studies, there was this large discrepancy between the in vitro findings and the in vivo findings. Okay, sorry, so that's a long-winded answer.
Great.
But I'll stop there.
Great, Pranita, for this comprehensive view. Robert, just following up on Pranita's comment, so you've worked on beta-lactamases almost all your career, and there's not many people who know more about those than you. So why do you think this is the way to go? Since there is very likely that in evolution, changes in these beta-lactamases are going to generate resistance very easily?
Well, that's true. It's a good question. I think, the best way to answer this is, when you look at our current landscape, we have about 12, 14 different combinations right now in development, which is a lot. And, as Pranita alluded, you have the DBO class, and then you have the boronic acid class. And there have been some ways to, a good one is taking a brand new cephalosporin, ceftolozane, and marrying it to an old beta-lactamase inhibitor, tazobactam. The enzymes are always going to change. How they change is a very interesting observation. In many bacteria, we know, in many enzyme systems like TEM and SHV, when they became resistant to the inactivation by clavulanic acid, they developed a weakness in their cephalosporin and penicillin hydrolysis. They became less catalytically efficient against the substrates that partnered with them. We know that now in the case of avibactam and in the case of relibactam, we don't have that clarity yet. As if you were, avi resistant, and if you were rel resistant or even Weber resistant, how much that catalytically impairs, because the resistance we've seen now, when isolates are ceftazidime avibactam resistant, they're usually, the mechanism for that is that the ceftazidime is broken down faster by the mutant KPC, for example, than otherwise. So it's different. I think the new inhibitors, it's going to be very resistance to an activation by these compounds is going to occur, but I'm not sure about their impact. I think the biggest impact is resistance to the partner beta-lactam, because if you become a beta cephalosporinase and you combine ceftazidime with avibactam, you actually break down more ceftazidime. And this was described in Pittsburgh by Dr. Ryan Shields and others, and it's been described in other locations where you can get ceftazidime avibactam resistance. Now, that could be overcome by imipenem alone or by meropenem alone, because the KPC has been significantly hindered in its catalytic ability to hydrolyze imipenem. So that's a way around it is to have a very good beta-lactam partner that if resistance emerges in the enzyme, the beta-lactam partner can't be overcome.
So, Pranita, and thank you for that great answer, Robert. I'm just thinking about my colleagues who are not experts in the field. And you are seeing all these compounds coming, and that creates a lot of confusion. Where do I deploy this? Where should I use this? I know we don't have time to go through each drug and do detailed explanations, but as a clinician, I want you to think about, in general terms, What do you think about you deploy any scheme, any strategy to deploy these compounds without having to call Robert or you all the time?
Yeah, so I think it's a very good question. I think for these agents to be used most effectively in clinical practice, there's probably a few steps to take. The first, I would say that if you are in a hospital where you have carbapenem-resistant organisms, and I suspect most people who are listening to this podcast are probably in that boat, I definitely think you should work with your stewardship team, your antibiotic stewardship team, to make sure that you have probably at least 2 of the 4 currently available beta-lactam, beta-lactamase inhibitors on your hospital formulary, potentially more depending where you practice. In my institution, which is in the mid-Atlantic, and we see a lot of resistance, we actually have all of these new agents on our formulary. I think you may get some pushback because people will say they're very costly, and of course they're more expensive than polymyxin-based regimens, but you should really remind people that we know polymyxins don't penetrate the pulmonary epithelial lining fluid well, which is for organisms like Pseudomonas or Acinetobacter where we see most infections, so that's obviously problematic. We also know their serum concentrations are suboptimal, especially when you're talking about patients with good renal function. And of course, we have several mostly observational studies saying for KPC producers, colistin-based regimens compared to ceftaz-avi is inferior. Our colistin-based regimens next to ceftold-tazo or imi-reli for imipenem-relibactam for Pseudomonas infections is inferior. And in all of these reports, what's consistent is there's greater toxicities with the polymyxins, particularly acute kidney injury. So I'd say step 1 is make sure you have these drugs accessible on your formulary. The second would be to work very closely with your micro lab, both because I think it's really key, especially with the carbapenem-resistant Enterobacterales, to know the mechanism of resistance. Knowing an organism is carbapenem resistance is just not enough anymore. For the CRE in the US, about half of the time, it's because of carbapenemase production, mostly KPCs, but certainly we see more and more of various metallobeta-lactamases and the OXA48-like enzymes. For the Pseudomonas in the US, at least for most regions, the carbapenem-resistant Pseudomonas, the likelihood of a carbapenemase is still relatively rare. But the big point here is that the way we would manage something that's NDM producing versus KPC producing is very different. So it's important to have some sort of testing modality to differentiate. And there's a lot of options available. For example, there's something we've brought on a couple of years ago, the Carbo5 lateral flow assay, which almost looks like a pregnancy test. It's cheap, it's very sensitive specific. So we can see within 15 minutes what carbapenemase mechanism is present to help direct therapy. And then, of course, I think in terms of the AST testing, it doesn't help to bring on new drugs. And this will become ever more important with some of the newer beta-lactam, beta-lactamase inhibitors if we don't have a way to test susceptibility. So I would encourage everyone to work with their micro labs and make sure there's some sort of method, preferably in-house method. And I would say preferably testing that's done in parallel versus sequentially. So for example, I know ceftold-tazo is not available to any of us at the moment, but if that's a drug that you usually use, I wouldn't say if you see a carbapenem-resistant Pseudomonas, just test that drug, because we don't know if it'll be active, and you really should be testing based on the drugs we currently have, other drugs like ceftazidime, avibactam, imipenem-relibactam, as well as potentially cefiderocol. And then the final thing I would just say is, once you get the drugs in the hospital, you get the formulary, it's really, for your average ID clinician, I would say, every hospital right now in the US has an antibiotic stewardship team, and they should be really responsible for messaging how to manage these. One thing is with all the newer rapid diagnostics we have, often what happens in a lot of labs is it might be reported a KPC is detected or an NDM is detected, and most clinicians don't know what to do with that information. So you certainly want to make sure clinicians know who to call when one of these is detected. And then I would say, just for the stewardship teams, as Robert mentioned, and as Cesar was saying, it's great we have these new drugs. Unfortunately, soon after they become available, we see resistance. So the classic example, he mentioned ceftazidime and KPCs. Another example might be ceftolozane, tazobactam, and Pseudomonas. And we know that the most frequently reported cause for emergence of resistance to ceftolozane, tazobactam is alterations in PDCs, or Pseudomonas-derived cephalosporinases. And these are chromosomally encoded class C beta-lactamases unique to Pseudomonas. And a lot of this work, of course, was done in Dr. Bonomo's lab. And the finding that amino acid substitutions, insertions, deletions are associated with toltase resistance has been shown almost in two numerous account publications now. And this most commonly occurs because of an E219K AMC variant. And this is when there's a negatively charged glutamic acid residue at position 219, which is changed to a positively charged lysine, RK. And this can lead to pretty dramatic increases, something like a toltazo MIC from maybe 0.5 micrograms per ml to 16 micrograms per ml. And the point of all of this is just to say that not only do we have these drugs, we might test susceptibility, but it's important to think about what should we do in clinical practice, knowing resistance can be common. So to take this back clinically, I would say that, and just to give you two examples of this, we had done some work looking at patients in our hospital with Pseudomonas infections treated with toltazo, and looked only at those patients who had subsequent isolates in the next 30 days of Pseudomonas. Obviously, there's a little selection bias in these patients, but half of them actually developed pretty high-level resistance to ceftolozane tazo in their subsequent isolate compared to the index isolate, and mostly due to these PDC variants. And Ryan Shields, who Robert had mentioned, had done very similar work at Pittsburgh that was actually published in this month's Antimicrobial Agents and Chemotherapy, where they actually found a staggering 80% of patients who had subsequent Pseudomonas isolates after being treated with toltazo had high-level resistance. So the reason I'm bringing all of this up is we need, in the antibiotic stewardship world, to help our clinicians. I think we also need to know not only is what's our sort of go-to drug, but what's our backup plan. So if we had a patient who had a bad Pseudomonas aeruginosa infection, they were treated with ceftolozane, tazobactam, maybe they went home, they're coming back, we think they're coming back with a failure of therapy, it's important to think about what the plan B here is. And it might be that, we know from lots of experiences now that cross-resistance between ceftolozane, tazo, ceftazidime is very high. So it might be that we think about a different drug such as imipenem relibactam, which has a different mechanism of resistance. And if it was active initially, it's very likely that it's still going to be active against the subsequent isolate, and it might be something to consider while waiting on susceptibility results. So I think to short answer to your question, Cesar, is I think as an average clinician, this field can be quite overwhelming because there's a lot of information out there. There's new publications daily coming out, but I would say that working with your stewardship team and working with your micro lab to make sure you have the drugs, have the testing set up, and then have some sort of general guidelines about first agents, and then if resistance developed, what the next step would be, is really important.
See, I agree with Pranita. I think what we sometimes fail to appreciate is that a pathogen like, say, Pseudomonas, has a very complex trajectory towards the development of resistance, whether they're under the pressure of a cephalosporin or under the pressure of a carbapenem. The trajectories are very, very different. When you treat a bug with a Pseudomonas aeruginosa with ceftazidime avibactam, you see an entire series of mutations that evolve not only point mutations, but also the enzyme is modified. There's sections of the enzyme that will go a 21 nucleotide deletion. I know that's almost unbelievable to even conceive, that you can actually, the enzyme, by a process of recombination and editing, actually, is missing 7 key amino acids. And, missing that 7 key amino acids confers significant resistance to cephalosporins. Mechanistically, it's very difficult to put your head around that. If you put an organism under carbapenem selective pressure, you get a different series of mutations. You get more entry-level mutations, porins, etc., efflux pumps. So the organism is, will respond in depending on the choice of your drug giving to that patient in a different way. And, as a result of that, the mechanistic changes that occur in the beta-lactamases are different. What makes it, cephalosporin resistant? Well, it develops a couple of mutations. All of a sudden, ceftolozane or ceftazidime, which can't fit there, fits in a lot better. And, the enzyme is able to, handle that substrate in a different way. The inhibitors, most of these mutations, when they occur, they make the beta-lactamase more susceptible to inhibition. But it's a balance. It's a balance between how much cephalosporin is allowed there and how much inhibitor is allowed there. And I think as we go forward, what will eventually develop is, just like Pranita said, when you have a bug and you have a resistance profile, you got to ask yourself, am I dealing with a metallo? Am I not dealing with a metallo beta-lactamase? Because that's a key point. And right now, we don't have drugs that cross over all 4 classes of beta-lactamases. We have drugs that cross over 3, but not all 4 yet. Pretty soon, we're going to get some really, potent beta-lactamase inhibitors out there, what they call ultra broad. Like Qpex is developing an ultra broad beta-lactamase inhibitor, and that's going to be able to inactivate all 4 classes of enzymes. And taniborbactam being developed by Venadarex. Again, a very oxyboron, a little bit same type of drug, not similar to, just same type of drug as vaborbactam, but taniborbactam is going to hit all 4 classes. So see, that's going to be a real challenge for us, because all of a sudden we're going to have a drug that a combination drug that's going to be very effective. How do we use them wisely? How do we do stewardship the best way? How do we do the right pharmacoeconomics? Because they are going to be expensive. It's going to be challenging.
Sorry to interrupt, but I just want you to follow on that, because I had a particular question about this ultra broad spectrum activity of these drugs.
Okay.
We are seeing, the field moving to there. Do you think that's the right way to go, or should we, chemically and evolutionary, should you think about more targeted approaches, because the cost of just being blind probably at the beginning will be good. You could use a beta-lactamase inhibitor, this broad spectrum, and then the clinician say, you see, I don't have to worry about class A, B, C, D, and all the beautiful things that Pranita just said, because I don't really understand it. So I had this drug that is going to hit them all, and I'll be done with it. So do you think that's going to be the case, or are these drugs really as potent, or it will be very easy to become resistant to it?
That's a 3-part question. Let me answer it one part from a point of view of, actually, being in medicine since 1986. You can't stop progress. And, we're going to develop these drugs, and these drugs are going to be used. So the fact that we already have these very potent drugs in the pipeline, we're not going to be able to stop that progress. Is that a good thing? I think so. I think that in no matter what instance you have, you will always have to balance therapeutic decisions, life or death decisions versus other decisions. And having something that will rescue a patient And, it's almost an ethical imperative to develop that. If you have the technology available to you, it will, and there will be benefit from it. Where I think the downside is that once you start abusing these agents and using them for indications that they're really not indicated for, or more laziness on everybody's part, or they're trying to dodge a bullet that's not really there, that's when you develop abuse. There's nothing wrong with the drug ceftazidime. Ceftazidime is not morally inept. It's a very potent cephalosporin. We overused it in instances where we shouldn't have probably used it, and as a result, we get ESBLs that have emerged, and we have all that. So the onus is not on the development of the drug. If we have the knowledge, we should provide best care for our patients. The onus is on how to manage the stewardship. So, but I'm in favor of the development of these drugs. I think we should develop them.
Sure. Pranita, one of the things that I heard from actually very respected people in the field is why would the clinicians not have the freedom to choose the combination? Why should now that these things are progressing so rapidly and we have a menu of beta-lactamase inhibitors, why can't we use with the particular cephalosporin? Why can't I use avibactam with cefepime or with other beta-lactams and so on and so forth? Do you think that's a good idea, or what are the limitations for an approach like that?
Yeah, I mean, I think, right now we're obviously at the mercy of what's available. There are not beta-lactamase inhibitors without beta-lactam partners, and I'm not aware of plans for the development of beta-lactamase inhibitors as standalone agents. And I'll actually ask Robert after this if he can speak to anything he knows about if there's stability issues as to why that's not possible. Right now, I think when we have beta-lactamase inhibitors co-formulated with beta-lactam, there's usually two considerations. One being that the beta-lactamase inhibitor should be active against the beta-lactamases capable of hydrolyzing the beta-lactam, right? So there needs to be a logical reason why it's being paired based on what enzymes hydrolyze the beta-lactam component of the combination. And the second is that there needs to be similarities in some pharmacokinetic properties, such as elimination half-life, metabolic pathways, biodistribution, to ensure that the beta-lactamase inhibitor is actually protecting the structural integrity of the beta-lactam, because obviously for the duration of the dosing interval, because of course if the beta-lactamase inhibitor gets eliminated very quickly and that beta-lactam is all by itself, then it sort of defeats the purpose of the combination. Right now I think we have the 4 beta-lactamase inhibitors that were discussed that are currently available to us, and hopefully there's several more that of course I agree with Robert that I'm excited will eventually be options for us, particularly when we think about drugs like Acinetobacter and some of these other organisms where and of course all the metallo-beta-lactamases where none of our existing beta-lactamase inhibitors are active. But one thing I just wanted to say is, while we're sort of at the current moment have these 4, I think we need to be very careful about making sure we're optimizing their use. So I'll give you an example. For imipenem relibactam, the package insert dosing is for people with normal renal function, 1.25 grams. So this translates to 500 milligrams of imipenem, 500 milligrams of cilastatin, and 250 milligrams of relibactam. But I would argue that if you're using ImiReli to treat a very drug-resistant Pseudomonas, you might want to think of pushing up the dose. So you're giving 1,000 milligrams of imipenem, which would translate to 2.5 grams of ImiReli. A similar example would be for ceftolozane tazobactam, where the package insert dose for UTIs, intra-abdominal infections, is 1.5 grams Q8 and 3 grams Q8 for pneumonia as a standard infusion. And I would argue if you're treating anything outside the urine, and obviously, if you're treating urine, make sure it's actually an infection, not colonization. But for anything outside the urine, that you want to give that higher dose, the 3 grams every 8 hours, and possibly as an extended infusion. And then, the last thing I would say with your question about the combinations, one combination that I think it's just worth mentioning, again, a lot of this work is from Robert's lab, is that we have this growing body of literature of the benefit of ceftazidime avibactam with aztreonam for metallo-beta-lactamases. This can be seen for Enterobacterales producing MBLs. So, for example, aztreonam can withstand hydrolysis from MBLs, metallo-beta-lactamases, but usually, for example, the NDM gene is on a plasmid with maybe OXA48 plasmid-mediated AMCs, CTXM, ESBLs, which can hydrolyze aztreonam. So partnering it with ceftaz-abi can be quite useful. And the second example would be for stenotrophomonas, which has intrinsic L1 metallo-beta-lactamases, as well as intrinsic L2 cephalosporinases, where this L1 metallo-beta-lactamases, chromosomal metallo-beta-lactamases, can hydrolyze, the penicillin, cephalosporins, carbapenems, but not aztreonam. But unfortunately, the extended the L2 can hydrolyze aztreonam. So basically, partnering the ceftaz-abi and aztreonam combination for these stenotrophomonas infections, and I'd say particularly for in the hem-onc population where we're seeing more and more of these severe infections, is another beneficial strategy clinicians can do when we're sort of confined to having the existing beta-lactamase inhibitors. So Robert, if I could actually ask you if you have any thoughts about why,
Yeah, the combination.
You
don't need Cesar anymore.
No, we've been in discussions where you have a really good beta-lactamase inhibitor, and Pranita's right, and Cesar, I know, has talked about this because, you've taken care of some very sick patients. You've always wondered, if this were paired with, let's say, for example, if this were paired with cefepime, could I offer my patients more than it being paired with imipenem? And, and I think, I hope I don't speak incorrectly. I think, one of the ultra broad spectrum inhibitors, QPEX compound right now, is not really paired with a specific beta-lactam. There's development that you could maybe pair it with the drug that you wish. But I think if that scenario rolls out, Pranita is absolutely right. It has to be given with the drug that there's a matched PK/PD profile. Even, when I was talking to Dr. Stephen Projan this past week, he mentioned that how much of the science is dependent upon how good that match is, so that the beta-lactamase inhibitor is protecting the beta-lactam during the dosing interval. And that's a very key consideration. How I think it's going to come out is that if you want to use this compound with this beta-lactam, you got to use 2 grams of this beta-lactam with 0.5 of this beta-lactamase inhibitor in somebody who's got a creatinine clearance of this window. So I think it's going to be very prospective. I don't think it would be wise to let us behave as if we were in the Wild West without any rules or regulations and just be cowboys and cowgirls and mix and match whatever we want. I think there's going to have to be probably prescribed guidelines as to how to do it to ensure patient safety, because I think that's the ultimate goal. You don't want to put patients at risk.
So the last part of the podcast, I want to talk a little bit about mechanisms. I love mechanism. So these newer beta-lactamase inhibitors seem to have a dual mechanism of action, not only inhibited the enzyme as their name suggests, but also binding PVPs and becoming sort of a beta-lactam on their own. So how much of that activity, Robert, you think that's useful and will make less likely for bugs to develop resistance?
See, I think that's an exceptionally elegant strategy. The reason I say that is when, as Pranita said, you had the ancestral protein, the penicillin binding protein, if you will, or the DD peptidase that was important in cell wall synthesis. And the theory is, or the hypothesis is, that the beta-lactamases evolved from these primordial enzymes that were important in creating cell wall. It makes logical sense that since a beta-lactam can fit in the active site of a PBB, that it also fits in the active site of a penicillin binding proteins. So optimizing that strategy of being able to inhibit a single or multiple beta-lactamases and at the same time being able to inhibit multiple penicillin binding proteins really accelerates the advantage on the side of the clinician. It empowers the clinician, if you will, because now you have multiple targets that you're dealing with a single drug. Nacubactam, Zidabactam, which is being developed by Wockhardt. Nacu was a Roche product. I think at one point it was a Roche product, then got sold off. But some of these those types of beta-lactamase inhibitors, Nacubactam, Zidabactam, offer an incredible advantage. And you could use them on their own. Theoretically, no, at this point, they don't want you to use them on their own, but you could, because they lower MICs dramatically. And I think that's a very clever strategy. And I think the synthetic chemists in the field really, when they're trying to make the DBO a better molecule, are looking to inactivate not only the beta-lactamase A, C, getting better at inactivating class D, because that's been one of the problems. With Vaborbactam is not a great agent against OXA48, but Avibactam is. Taniborbactam is very good against OXA48 and the others. So the synthetic chemist has got real challenges ahead in being able to do cross-class inhibition. And I think it's not only cross-class of beta-lactamases, but the multiple penicillin binding proteins that you could get with a single molecule will also be extremely beneficial. That's going to result in sidality, and that's what you want. You want a rapidly bactericidal antibiotic that you could use in patients that need it.
So I'm going to give Pranita the last word, and I want you to put your futuristic view, the crystal ball for the future when these compounds become available. Are you optimistic? I think this gives us some bright light that we're going to have drugs against these bad bugs, particularly Acinetobacter and Pseudomonas. So how would you envisage the world would be post-pandemic and new drugs? How? A happier place, right?
So I think the only thing we can say with confidence is that for those of us interested in the mechanisms and treatment of gram-negative resistance, that we're going to have some job security for some time, because this is not going to be an easy issue to solve. I'm very excited about all the new agents that will be available to us, especially, as Robert mentioned, with some of these newer drugs, cefepime, Zidabactam, and Mirapenem, Nacubactam, cefepime, Taniborbactam. So some of these drugs that are a little less are more indifferent to the serine versus metallo-beta-lactamases. I think it might, as you said, cause some academic laziness on the part of clinicians when they don't necessarily need to think about the mechanism. But at the same time, I think it means, unfortunately, once we get to a point where we're using some of these drugs empirically before having results back, AST, culture growth as well as ASTs, it will ultimately help the patient. But I also think, I think about something like carbapenem-resistant Acinetobacter, where we know that it generally is producing lots of OXA-type carbapenemases, OXA23, OXA24, OXA58, OXA51, if there's a specific insertion sequence. So we do one of the newer drugs, the Selbactam, I think it's Durlobactam. Is that thank you. Where we know that high dose Selbactam is saturates penicillin binding protein 1,3, So it can be quite effective against Acinetobacter, but having this beta-lactamase inhibitor will help with some of these oxocarbapentamases, hopefully, if what we've seen so far with this agent pans out. But, we also know that carbapenem-resistant Acinetobacter can produce metallobeta-lactamases sometimes, right? It can produce New Delhi metallobeta-lactamases too. So I think that despite some of these seeming, quote, ultra broad and so forth, there's still going to be gaps as new carbapenemases that have not yet been identified emerge, as well as some that we don't commonly see in certain organisms become more prevalent. So I think that the field will be exciting. My guess is that in a few years, we'll sort of be back to sort of where we are now, where we have more options, but we're also going to see more resistance to these drugs. And then I just wanted to say one quick thing. Just being a pediatrician, I think I get a lot of questions from pediatricians about how come we don't have pediatric agents. And the one thing I'll say is the FDA has done a very nice job of making sure that when a sponsor is submitting a request for a new agent, that they do have a plan in place for a pediatric development program, usually within about 60 days from the end of the phase 2 study. And there's some nuances that exist there. But from my point of view, if we have good in vitro susceptibility data, we have good animal data, we have safety data in adults, there's never a reason we shouldn't be using a drug in children. We're often used to using it off-label. And certainly, feel free to reach out to me if there's questions about some of the dosing considerations. But I personally think as a pediatrician, there's no reason we should approach the use of these agents any differently than we would with adult patients.
Fascinating conversation. The first thing I need to do is to learn how to pronounce all these new drugs. But I'm also, as you, very excited. I'm very thankful and grateful for you guys for this fascinating podcast and very insightful.
Thank you, Cesar.
This is Cesar Arias, Editor-in-Chief of AAC, signing off.